Good afternoon, everyone. It's a pleasure to be speaking to you today, and I wanted to thank the Post-Harvest Technology Center for throwing this event, and Erwin and Angela and everyone who helped throw it on. My name is Mitchell McCartney, and I'm the Director of Research for the Biomems Lab here at UC Davis. That's under the faculty supervision of Christina Davis. Christina is both a professor in mechanical and aerospace engineering, and currently she's serving as the Associate Vice Chancellor for Interdisciplinary Research and Strategic Initiatives here on campus in the Office of Research. I'm a staff member here on campus. I've been working with Christina for over 9 years now. My topic today is going to be related to the use of VOC sensors to help with assessing quality in produce. So today in my presentation, I'll kind of give you a quick introduction to our lab specifically and some of the technologies we've been developing. I understand it might be a mixed audience among us, so I'll do a brief introduction about VOC specifically to define what they are and some of their roles in biology. And then I'll focus on some discrete studies that our group has led with the fantastic support of collaborators here on campus, some of them among us. And I'll highlight some of the ways that we've demonstrated VOCs to be used for produce quality. Specifically, I'll talk about mold detection in strawberries, used VOCs to classify walnuts by grade. I'll talk about some agricultural pathogen detection that we're doing, especially related to citrus greening disease. And then also the use of VOCs in fermentation monitoring. So maybe I'm stretching the definition of produce a little bit, but I think it's pretty applicable to talk about like an inline production sensor in there. And at the end, I'll kind of just go over briefly, depending on time, a little bit about more detailed about some of the technologies our group is working on today. So officially, our name is the Bioinstrumentation and Biomems Lab. We go by Biomems as a brief name. There's 27 of us in the group between staff and postdocs, graduate students, and student assistants. And our group really is divided into two different parts. We have, of course, our engineering team, who is really focusing on the sensor development. And we're really interested in no longer needing to bring samples into a laboratory, but really bringing the laboratory to the point of sampling instead. So we're working on more mobile platforms that have appropriate sensitivities, for their application needs, and really get technologies into the hands of people who need them. And of course, since we do a lot of custom sensor development, we have a lot of custom data. And we always want a minimally trained operator to be able to use our technologies. So we also incorporate a lot of software and AI, ML algorithm development so that these devices can automatically process their data and output simple response. Now, in developing these chemical sensors, we need the help of our chemistry team, both to help the engineers understand some of the design criteria to perform chemical analysis in these technologies. But also we do a lot of gold standard instrumentation using a lot of traditional benchtop laboratory equipment just to help with some biomarker discovery and also to find, maybe to do some proof of concept studies to find new applications before diving into developing a specific technology. So some of the toys, if you will, that we've been working on, actually one of the primary research areas for us is breath research and developing new health diagnostic tools based off of compounds that you exhale. So our engineering team has developed a number of ways to collect breath samples. Let's see if I can turn both for humans, and also we've actually done some breath work with dolphins with the Navy down in San Diego. Of course, chemical sensors are a big part, working more towards lab on a chip, small portable devices that we can move around. And because we're trying to miniaturize so much, that involves a lot of microfluidic chip development. So we do a lot of work in the clean room here on campus to make really small scale etchings in certain substrates for our sensor technology. And I mentioned, of course, algorithms and software to automatically go through the data that we generate, provide an answer. And we're also looking at different ways of capturing samples. So we have personalized air sampling techniques I'll go over later, so you can monitor personal exposure to VOCs, for instance. And we've also done some work. We've put VOC sensors onto as a payload on a drone for some field applications. And I'll talk about that later. Our chemistry team, what I'll be talking about today is really pre and post harvest monitoring and using VOCs related to that. We've also done some work for the animal health. I mentioned some dolphins. We're actually doing a study looking at the microbiome of cats as part of their diet. Breath research, another big aspect of our team. And then something else I'll present today is the fermentation studies. So quite a number of these I'll be going over in detail as we move on. So what are VOCs? I mean, VOCs just stand for a volatile organic compound. And essentially, they're just carbon-based chemical compounds that exist at room temperature in the gas phase. So they can readily evaporate, and some concentration of them would exist here in the air. And of course, they're responsible for what we perceive as odors. So as the volatile compounds interact with the olfactory system, our brain interprets that as some sort of smell. Some VOCs smell fantastic. Glasses of wines, those contain dozens to hundreds of different individual volatile compounds, and the combination of those is responsible for our sensory perception of that glass of wine. And of course, a bouquet of roses, certain varieties at least, also smell great. And then, of course, I think VOCs tend to have a lot of bad associations with them in a lot of consumers, because we think of them as maybe off-gassing from paint or the products of industry. And of course, there are VOCs that have very serious health concerns. A lot of those can be formed in combustion, so wildfire events, smoking a cigarette, those can generate some volatile organic compounds that are very dangerous for us to inhale. But certainly there are hundreds of thousands of different volatile organic compounds, some of them great, some of them not so much. And all living things emit volatile organic compounds. And some of that may be active, in which there's a purpose for them to be off-gassing these compounds, and some of it actually may be passive, where inadvertently these compounds just end up being released. And so living systems have hundreds of thousands of different metabolic biochemical reactions occurring. And the end result of a subset of those generates volatile organic compounds. And so some examples of those obviously would be humans. We use pheromones as terms of mate selection. And other animals, I'm walking my dog around the neighborhood. He loves to leave behind his individual scent marker all over the place as a form of communication. And even in bacteria, there's examples in which they are releasing VOCs as sort of chemical cues for proliferation, where they can kind of speak to each other as a colony of bacteria that way. So related to produce, there are plenty of examples that have already been documented examples here about VOCs serve very specific biological functions. So here's a case where a plant may be off-gassing actively a VOC for a reason. And in this example, we have a plant-insect communication going on for some plant defense. So here we have some kind of plant that is under attack by this herbivore who's munching away at the leaf tissue. As a result of it being fed on, this plant will end up releasing some volatile organic compounds, methyl salicylate being a classic example of this. That's actually sort of a stress signal that it's emitting. And it turns out that through evolution, these compounds end up attracting an insectivore that will then come attack the herbivore. So, the plant is attracting the enemy of its enemy in order to get some help to defend itself while under attack. I think another more fascinating example of this in biology, especially plants, is this communication can occur between plants of different species. So there's an example of a sagebrush when under attack by another herbivore here. It, similar to the example before, will release some more VOCs. But it turns out that a plant of a different species, in this case wild tobacco, can actually sense these signals that the sagebrush is off-gassing. And studies have shown that as a result of this wild tobacco can end up actually building up natural defenses, so that when an herbivore would attack it, it would find it less delicious and actually there'd be less herbivory on that plant. And also it can start to build up defenses such as building up more seed capsules in the event of mortality, at least it can continue on. And so these are just two examples, but there are so many more in which there is so much communication going on in plants, especially around us, that we are just not, our human nose is not perceptive to at all. In terms of animals, and human smell is the smell of humans. Our ability to smell is notoriously weaker compared to many other animal species, and so we're really unaware of so much going on around us in this regard. So those are just some examples, but there are so many others in which VOCs can be altered. Again, VOCs are the end products of certain metabolic pathways. So you can imagine there's many different stressors that can then alter the types or the abundances of VOCs that living systems, plants, produce generate. Biotic stressors, we talk about pathogens, competitors, pollinators, herbivores, but abiotic stressors too, you have temperature, humidity, radiation, nutritional deficiencies. And I'm sure most of you are already aware that all of those can also impact produce quality. So there are already so many mechanisms that can end up having an altered VOC profile in a system that might very well be related to something of a concern to a producer. And so that's really the idea behind so much of the technology our group has been developing, is that you can identify some kind of volatile fingerprint related to whatever it is you might be interested in terms of produce quality, for instance, and that you could then tailor a chemical sensor specific to detect that chemical signature and train it to be able to identify it. And then with the right different algorithms to be able to interpret that data and to correctly make a prediction. Again, you want minimally trained operators to be able to use this technology. You could have a system put out a really simple, basic, response to whatever it is you're trying to measure. And I've heard this actually came up in the break that some groups have worked. There's a technology called e-noses that some groups have used before, and I have actually never used them myself. I just want to be clear that none of the work I'm presenting has used e-noses. They have kind of a bad rep to them. So if you've worked in this realm before, I encourage you to continue to listen with an open mind, because there are different alternative sensors that are certainly fantastic for VOC measurements. So switching gears and now moving into some specific examples that my team has worked on. This is actually an ongoing study we have now, and this related to strawberry post-harvest quality. Of course, as a consumer, the worst is picking up strawberries or blueberries or figs, and the next day they're moldy, right? So it's a common occurrence here. And of course, one causal agent of this might be botrytis, especially in strawberries, which is a type of fungus that can generate this type of mold that we see. Fungi and bacteria and other microorganisms, they're kind of stinky. I think one of the greatest ways for us to make sure that the food at home is okay to consume is you smell it, right? You take your milk carton and you give it a whiff, right? So that's actually to the benefit of these VOC sensors, that a lot of these pathogens, produce unique signatures that don't exist in these plant species that are identifiable. So of course, the question is more so, how early could a VOC sensor detect rot? Could we detect it before visible symptoms? Could in a packaging plant you be able to, quarantine off a certain, inventory because it shows signs of this early on? I should mention this is a collaboration with Professor Barbara Blanco Alate, who is in plant sciences here at UC Davis. So how do we measure the strawberry VOCs? Well, we're using a technology. So again, this is using a gold standard laboratory benchtop technology called gas chromatography mass spectrometry. It is by large and far the gold standard VOC measurement technique. So we took strawberries, healthy and inoculated, and this grotesque picture, I should mention, they did not have visible signs of botrytis at the time that we were sampling. And we also did this work with 3 different varieties of strawberries: Monterey, Rolls Royce, and Fronteras. And we've essentially put the strawberries into like their clamshells, and we use these. These are called twisters. They're essentially VOC sponges. They're wrapped in a chemical absorbent that essentially, as they're in the container with the strawberries, they quite literally absorb all of the volatile aromas within that container. So it's kind of pre-concentrating and collecting a snapshot of the VOCs at that time. And then we analyze those twisters using gas chromatography mass spectrometry. And so after inoculation, we had measurements at 5 different time points to follow sort of a time course. Now, if we don't consider the time point that the measurements were made. We saw pretty good accuracy to be able to predict, based off of VOCs, which strawberries actually were inoculated with the rot. And so on this left plot, you're seeing that the strawberry samples, each one represents a single VOC measurement we made, and they're colored by what we knew the inoculation status to be. So there's almost nearly a diagonal line you could draw between these that would really completely discriminate between the strawberries with rot and strawberries without. And overall, the ability to correctly identify inoculated strawberries was at 86%, and to identify healthy strawberries was at 85% accuracy, irregardless of the time point. And what's interesting here is that this middle plot is the same plot on the left, except for instead of coloring the samples by their infection status, I'm coloring them by the strawberry variety. And this is showing that using our chemometric techniques, that we were able to identify between these three varieties a universal signature of this rot. So it wasn't necessarily specific to a certain type of strawberry, that there was some sort of universal VOC fingerprint for botrytis. And then when we went back through and looked at the specific time points, we collected samples of strawberries 9 hours after inoculation, 12, 16, 20, and then up to 24 hours after inoculation. And of course, the accuracies did change between time points. And of course, we always see in a lot of our biological studies, there's also living systems, that have their own circadian rhythms a lot too. So their VOC production does change over the course of the day. But nonetheless, we saw that the accuracy still remained between 72 and up to 92% to identify, to discriminate between these, to identify those with the rot, and that our strongest time points were between 16 and 24 hours after inoculation, which were certainly far before any visible symptoms were seen. So you can see how this could be especially beneficial to the storage of strawberries to be able to start quarantining or removing those that show signs of botrytis. Now, using GCMS, we get a lot of information about what compounds are exactly being used for the models to distinguish between these two sets. So we had a total of 131 volatile organic compounds that we're measuring in these samples. Here's a select list of some of those. So you can see it's not just a dozen chemical compounds that are being emitted, it's hundreds. Now, in terms of the biomarkers of the fungus, I mentioned they're stinky, so some of these probably were directly related or directly came from botrytis, but also there is an interaction as the strawberries are defending themselves. They're undergoing new biochemical processes, and thus their volatile profile is likely changing as well. So it's probably, in this case, a combination that the model is using both fungal VOCs and strawberry VOCs as a response to infection. And so this type of work is important because this helps us in engineering to later understand exactly what volatile compounds might be needed so that we can tailor custom sensors to those types of compounds. But an interesting kind of result of this work too is that, again, these are end products of metabolism. So you're starting to maybe reveal some biochemical or metabolic pathways which might be able to, as an opportunistic way for some other researchers to look at potential preventions or therapeutics, since you're seeing those that are directly affected by botrytis. And another work that we have ongoing now is using VOCs to be able to classify walnuts by grades. So I'm a big fan of walnuts, but they go rancid pretty easily. They contain healthy polyunsaturated fatty acids, but of course those are pretty prone to oxidation and thus turning rancid. And of course the quality, that quality loss certainly impacts product desirability, storage, its potential for walnuts to be used in other snack foods or other commodities as well. And so of course walnut grade could be determined by smell. You could go through and smell walnuts and get a sense of how rancid they are. But of course, that's not an extremely high throughput method. There's olfactory fatigue concerns as well of people going through and just, as we get exposed to certain things, naturally our senses just become a little bit more numb to those. And of course, there's subjective qualities there as well. So the question here was whether VOC sensors could be used to better classify walnuts in terms of a grade of rancicity. And this is actually with Professor Elizabeth Mincham, who's here and is the director of the Post-Harvest Technology Center. So what we've received samples of walnuts where their rancidity grade was previously determined by somebody else, by Beth's group. And so we have samples where they're graded on a scale of 1 to 7, 1 being sort of the freshest, 7 being the most rancid. And I'll mention, this work is certainly ongoing, so I'm presenting some preliminary results. This is funded by the California Walnut Board. But essentially, to make the measurements, we put walnuts into a container, just a simple vial here. There's a septa here that we can connect tubing. And in this case, we're using a technology called GC-DMS, differential mobility spectrometry. If you've gone to the airport and you've had them screen your luggage or your hands for explosives, sometimes they take a white pad and kind of rub it on your hands or your bags and run it through a machine. That's actually a type of DMS technology. It's a form of ion mobility spectrometer. So these are commercial instruments that are actually primarily were developed for explosives detection, but they have many different applications. And these technologies are great because they're much more amendable to mobile applications as well. They don't require as much ancillary equipment as traditional mass spectrometry. So we opted to use this in this particular case. So here you're seeing 4 GC-DMS spectra of walnuts sampled at different grades. So we have, in this case, we went from grade 1 to grade 4, so from the freshest to the most rancid. And in these spectra, each sort of peak or each blob represents an individual volatile organic compound that is being measured from the walnuts. And as you can see, moving from the freshest to the more rancid walnuts, you sort of start to see 2 things occurring. One, some of these peaks show up consistently in all of these samples, but their intensities increase. They're becoming more abundant as the walnuts were more rancid. And there's also a lot of examples in which VOCs are appearing that didn't exist in the fresher samples before. So there's already a great deal of chemical information here that we could use to be able to classify walnuts. And so again, if this is plotting the intensity of this peak, this first peak in these 4 grades, we saw a linear correlation with that grade, that as the walnuts were classified more rancid, the abundance of this particular compound also went up. So what we're working on now is developing some AI ML models. Erwin, I appreciate your comments about artificial intelligence earlier. It won't solve all of our problems, but it's an incredibly useful tool, especially in situations where the data streams are so complex. We need the benefit of some algorithms to do the mathematics for us instead of just a human. And so we trained this model on samples of these different grades, and then we fed in some unknown samples where we knew the right answer, but we wanted to see what the model would predict. And in this case, the model 100% with 100% accuracy could differentiate grade 1 and grade 2 walnuts. And then what we saw was it did conflate between the grade 3 and grade 4 samples, that as the walnuts were becoming more rancid, it was a little bit more difficult for this model to discriminate between those. But still, you could see how this could be fantastically useful in a sorting facility to be able to have, a premium version of a product versus, a good version of your product versus one that might be used for something else. And I should mention here, there's no destructive sampling here. We were just sort of measuring the walnuts whole. We use mass spec again because the mass spec gives us a lot of great deal about chemical information, identities of what we're seeing. Again, if you compare the volatile profile of fresh versus rancid walnuts, as with the DMS data, we saw a lot of compounds going up in abundance. We saw the formation of some new compounds. And again, in this particular case, the rancicity is caused by the oxidation of polyunsaturated fatty acids, which generate a whole slew of unique volatile organic compounds. So this was a pretty great opportunity for a VOC sensor to be incorporated. And there is a difference here, I should add, between these chemical measurements and the sensory perception of rancicity. That's a different work to be able to understand exactly what specific concentrations would it take for a human to be able to differentiate between some of these grades. Just to keep that in mind, since the human nose obviously is a lot less sensitive and a lot more subjective than this technology. Really quickly, as a bonus slide, we were interested too in looking at a way of sort of classifying another produce by grade, in this case, bananas by their ripeness. So currently, bananas are among the most shipped commodity in the world, and they're shipped under constant refrigeration just to make sure that they'd rather show up pretty green and ripen sort of at the marketplace rather than in transit. And so that's an incredibly energy-intensive endeavor. A typical 20-foot container uses about 55 kilowatts per hour of energy per day. So that's it's a lot of energy. So the question is, do you need to ship bananas constantly under cold conditions, or could you kick in refrigeration when you start to notice that they're ripening? So we did some preliminary work where we measured VOCs from bananas as they were ripening, and you could see that the volatile profile changed. And so you can imagine a feedback loop where a VOC sensor could be built into these shipping containers, and as it saw signals of ripening, then you could turn on the refrigeration, or at least maybe moderate it to what's only needed. And I think, of course, when everyone thinks of bananas and ripeness, they think of ethylene. But just to add in here that, of course, ethylene is directly related to the ripeness, but it's not the only volatile organic compound that we were using in this classification. So there's many others. So now moving gears a little bit and talking about agricultural pathogen detection. We've done some work using VOCs to be able to diagnose a specific citrus disease, citrus greening disease, also known as HLB. For those who haven't heard of it, it's the most serious citrus disease we've ever faced here in the United States. It's an invasive bacteria that's spread by an invasive psyllid, an invasive bug. So we had some plant material imported from China in the late 2000s that had the psyllid with the bacteria, and it has just exploded in this country. Florida used to be the #1 citrus growing region in the country. They are no longer, and it's not because it's actually California now. It's not because California got better, it's because this disease has decimated their industry. It's really unfortunate. It's spread, it's in Texas, it's also in California, but for some fortunate reason, the disease hasn't really taken off here in this state. So fortunately, it's still being held back at bay. But it's massively damaging to fruit quality. Essentially, what this disease does is bacteria fills up the phloem of the citrus tree with starch, and it basically starves plants of nutrients. There's a 1 to 2 year period between getting the bacteria and when plants show symptoms. So there's a very long latent period. It's really difficult to identify early on. And as a consequence, a lot of the fruit becomes, it impacts the visual quality, it impacts the flavor quality. You get some misshapen fruit, some bitter fruit, and eventually the tree just dies. And there's currently no cure for this whatsoever. And it's also really interesting to diagnose because, I think everyone's now familiar with PCR after COVID. This bacteria doesn't spread evenly in the tree. So you can pull 20 leaves from a tree and only one of them might contain the bacteria. And PCR needs the bacteria present to be able to identify the infection. So we wanted to look at using VOCs as a way to assess whether or not there was a host response to infection, looking at the citrus tree itself. Now, what's really interesting is an alternative VOC sensor has been demonstrated to identify HLB-infected trees with incredible accuracy. This is work by Tim Gottwald, who is retired, but he's, well, lightly retired, I guess I should say. He's worked at the USDA for a number of years. He's worked in training canines for pathogen detection in agriculture. So there were 10 dogs that he had traded. Of course, you have to show photos of the dogs. And he went through and the dogs got trained to be able to identify an HLB-infected tree. And this was a blinded study that was conducted over the course of 32 months, where they took citrus trees and they, of course, had healthy controls that were never inoculated, and then trees that they purposely inoculated. And what's interesting here is this black line is representing the accuracy of the dogs to identify HLB-infected trees versus some of the different PCR techniques that they were using. And early on, as little as 7 months after infection, the dogs had an accuracy rate of over 90% to correctly identify infected trees, whereas the best PCR version that they were using had an accuracy rate of 30% of the time. So just an incredible, incredible improvement to be able to find trees that are infected so growers could remove them before the bug ends up spreading the bacteria further. Of course, canines and their noses, these are just a VOC sensor. That's all they're doing. They're picking up these volatile compounds that these trees are off-gassing to make that distinction. It's not based off of visual, because many of these trees, especially at that point, are asymptomatic. They don't show any visible signs of infection. Now, dogs are great, and I love mine, but of course, trained dogs are expensive, and of course, there's some throughput issues here, but it just shows how other people working in this field have also demonstrated this ability for VOC sensors. So using the DMS, the technology I talked about earlier, this was work done in Florida. We ended up putting one on the back of a Kubota. Here's the device on the back. We have a power inverter and some solar panels to be able to run it through the field. And we basically just went up and started to capture some aroma profiles of these trees. Citrus trees in Florida are pretty big. They're over 2 meters tall. They're kind of more like a bush, so they're big. They kind of also maintain their own sort of, it's not a lot of air exchange going on, that they kind of like concentrate their own aroma profile kind of underneath that brush. So it's pretty easy to capture a VOC snapshot of them. And so using the DMS, we determined the volatile fingerprint of infection, where we saw examples of what healthy trees would look like and compare that to some infected trees. And we're able to pick up the biomarkers of infection. Similar to the dogs in a controlled greenhouse experiment, we proved that the VOCs could identify infection earlier than PCR was able to. And we did a time course study over a year where we kept sampling trees out in the groves in Florida. And overall, we found an accuracy rate of about 90% to be able to identify, so which aligns really well with what the dogs were showing. I should mention one of the hardest things for us was actually sampling in the springtime, and that's because this is when these trees in Florida were flowering. And I don't know if you've smelled a flowering citrus tree, but they smell great. But that introduced quite a bit of noise that did have some limitations and hurt some of our ability to make the diagnosis. And what's interesting then was another group, based off of our results, we identified, we were able to describe the volatile organic compounds of an infected tree. And so a collaborator at the University of Florida, this is Professor Stolinsky, actually working with the psyllid, the vector of the disease, put up like an olfactory tea maze where he released psyllids, and on one end they could go to a very delicious citrus tree, and on the other end was a cotton wick with our synthetic blend of HLB, if you will, from what we saw in the field. And it turns out in this study that our synthetic HLB blend was more attractive to the psyllid rather than a healthy citrus tree. And there's kind of two takeaways here. One, inadvertently, this shows how the VOC technology ended up discovering a new lure to help control the vector for the disease. And two, it really kind of shows some of the fascinating evolution of the disease, where it's actually making trees smell better to the vector so that they'll come pick up the bacteria in order to help spread it onto other trees. So it's pretty fascinating. Currently, this work is very much still ongoing. We have funding through the USDA NIFA in conjunction with the CRDF and Bayer, where since PCR, again, as fantastic and as sensitive as a detection technology that is, it's not accurate in this specific use case. So we're using our assay to help Bayer and some other groups go through and assess potential therapeutics and treatments for the disease. Again, I'll mention there is no cure for this disease. It's really devastating for the growers in Florida. They've been trying to deal with this for over 10 years now, and it's really unfortunate to see the consequences on the industry. So using a version of this assay we developed, we're helping that screening. And I had the fortunate ability, we can travel again, so I got to go to field site in Florida to actually go see some of these trees in person just 2 weeks ago. And so finally, in terms of an example, I'll talk about some VOC sensors in line for fermentation production. This work actually was in conjunction with, at the time, GE Healthcare. They're now Cytiva. And they do a lot of what they call bioreactor production, which also is just known as fermentation. And a lot of T-cell therapies for cancer patients. They're actually taking T cells from patients, doing some modifications to them and injecting them back into the patient to try to manage the cancer. And also this relates to production of different hormones and pharmaceuticals. but as well as fermentation in terms of beer and wine production, et cetera. So currently, the only way to really assess how well your fermentation is going is to actually quite literally pull a sample of it out and run it through some biochemical assays. And it turns out that this is pretty dangerous because this one little piece is where you can really accidentally introduce some pathogens into a bioreactor, which of course these are prime conditions for microbial growth. So those contaminants will just take off. And according to Cytiva, for these particular cases, there's about a 10% failure rate for bioreactors due to contamination. So again, the question was, could we use some sort of VOC sensor to help give some more information on just how the fermentation is going, if these cultures are happy and healthy and proliferating? So how to do the measurements, we wanted it to be non-invasive. That was the most important part. We didn't want to have another risk of introducing more pathogens. So in this particular case for the bioreactor, this was an inline sensor where this was an aerobic fermentation, so there was a supply of air going into the bioreactor, and of course an exhaust line. So we took off the exhaust line measurements, and again used these twisters, these VOC sponges, just to collect the odors of these bioreactors over a time course. So non-invasive, non-destructive sampling, no risk of contamination. And for this proof of concept, again, we were just using classic GCMS. So we retrieved the samples and brought them back to the laboratory for analysis. We followed these bioreactors over about 8 days of fermentation. And this is during the growth phase of these particular cells. We had 2 different types. This is, again, related to human health, but pretty applicable here, I think, to this industry. We were looking at CHO cells and T cells. And again, each dot here is representing a VOC sample that we took, and I've sized it by the day of the experiment. So as the experiment went on from days 1 to 8, and as the cells were growing and expanding, we certainly saw a shift in the volatile profile. And you can see that there was certainly a difference between the purple T cell samples and the green CHO cell samples. So each cell type had a unique profile, and that they both also shifted as they were proliferating over time. Now, using these VOC signatures, we asked the question, could we predict the concentration or the density of cells inside the reactor at the time? So as we were making our VOC measurements, they were going through and doing their normal biochemical analyses. So we had the exact concentrations of cells for each time point that we measured. And using some regression models, we saw some pretty decent accuracies for this proof of concept work, where for CHO cells, we could correctly estimate the number of cells with about 72%. There was a linear correlation about 72%, and for T cells about 84%. Overall, when you ask the question about error, it was less than, in all cases, it was less than 20% error to correctly identify, to estimate the concentration of cells inside the reactor just based off of VOCs in the exhaust line. So what's coming next? Where does this go? I've kind of done a mixture here of some different technologies using gold standard equipment. Earlier on, I mentioned some of the novel technologies our group is working on. I'll mention, where our technologies are most mature are related to human health and diagnostics. We have ongoing work from the NIH now to develop a breath sensor to identify COVID from a breath sample. The FDA actually authorized about 3 weeks ago its very first breath test for COVID-19. So it shows you there are products now emerging on the market to do exactly what we're doing. And again, these are not technologies looking for the virus. They're looking at the human's response to the virus and looking at measuring volatile metabolites to be able to identify infection. So some of the technologies I mentioned, we have a patent here. This is a micropreconcentrator, which is one of the tools that we use to capture VOC samples. It's sort of a small chip, about 1 inch by 1 inch. It's filled with a chemical sorbent that VOCs stick to. And when you heat it, you can get them back out. So you can kind of pre-concentrate a sample. I mentioned we put them on some wearable devices that people can use for personal exposure. We could put that in line. We've actually done some work in the egg industry that I'm not allowed to talk further on using this. I mentioned payloads on UAVs. You can imagine doing maybe an initial screening among an agricultural field just to see if maybe a pathogen might be present, and then you can go in more closely and try to pinpoint exactly where. And then we have another patent here on some of the new versions of the sensors that we're working on. So you saw the suitcase form factor. That was an earlier commercial version of our device, and we want to make them even smaller into something lab on a chip based. So we've developed a subset called half IMS, which is just another way of measuring VOCs. I think the UAVs are pretty cool. We've talked about putting samplers onto payloads as drones to do some VOC sampling. Since we are interested in human health, we actually did some measurements of VOCs here in California during the horrific Camp Fire in 2018, which was the deadliest wildfire, unfortunately, here, I think, in human history. And we went through and actually were able to capture and understand some of the volatile compounds that were in the air. I think when we think about air quality, we often think about particulate matter, but again, benzene, xylene, there are a number of volatile organic compounds that are extremely bad for human health. And we certainly saw those in the air during that wildfire event. And we're also able to use this in a controlled sort of, a simulated fire to also make some additional measurements about what VOCs are being released. And we did some experiments with the Department of Defense too, looking at being able to detect VOCs of concern in air. We had some small work too with Intel looking at, again, we were trying to develop lab on a chip. So can we get these technologies into where they really need to be used in a production line, in a sorting facility, wherever that may be. So we did some work here at that point that was geared towards maybe a VOC sensor on a cell phone, for instance. Again, just merging all these different technologies together. I'll mention our group. There's a UC Davis affiliated startup, Sensent Ventures, here in Davis, California. They've licensed a lot of the technologies, and they're working on commercialization of our hardware and software components, especially for our COVID-19 breathalyzer that we've been working on. And I'll mention here for those in industry who maybe are unfamiliar, our intellectual property, patents. These are all things that are, our innovation access office here on campus will work really closely with you, especially in the contract phase of work, to really be able to preempt and identify exactly who owns and who has rights to certain IPs in terms of the production of these technologies. So, I want to, campus definitely encourages us to be on the lookout for those as well. So I think I'm a little over, but in summary, VOCs for sensing produce quality. I've shown some examples here of just what our group has done, but certainly we're not the only ones working in this field. And I'm pretty excited, I have to say. I've been working with Christina for almost 10 years now on this, and I've seen so many examples just recently of some VOC technologies really actually becoming commercializable and hitting the market. So this is not an area that's exclusive to academic research laboratories for proof of concept studies. There are real technologies being developed and marketed out there for specific needs. So in terms of produce quality, we have demonstrated some non-destructive, non-invasive, reagent-free chemical sampling so that these sensors could be incorporated into mechanisms already in place. And it provides some actionable information related to identifying contamination or pathogens, related to being able to classify the quality of your product, and also to potentially help reduce with some manufacturing or some shipping costs. So, and again, there's so much possibility here for sensing technologies to be specific to industry needs. Our group in particular doesn't see this as where we will develop one device that gets used everywhere. We see this as really working with industry partners to really design and incorporate technologies specifically into where they're needed, and not having a universal device, so to speak. I have to, of course, thank many of the funding agencies who have been supportive of my team's work over the years, especially here related to Protos, the USDA NIFA, the California Walnut Board, Bayer, and the CDFA. And so I just wanted to thank you so much. I'm happy to stick around for how long I have for questions, and I'll be part of the panel discussion later too, in case we run out of time.
Maybe I missed it, but could you talk a little bit about the time factor in terms of the sensing and detection? Say, what would be an example? Say the citrus greening disease, or well, anything. What's the time there for detection and useful data for making a decision?
Yeah, and I think this comes up a lot in kind of designing this, where I think rapid high throughput is always the objective here. And so it really depends on the application, because some of these volatile compounds are emitted at such low concentrations that you do need to capture enough of them to be able to measure them. So there's limitations there. But some produce, as you know, we saw with strawberries, we saw with walnuts, we see with bananas, are emitting hundreds of compounds at pretty deep that are certainly easily measurable, even though they may not be sensed by the human nose at those levels. So in this banana case, we were doing just a 15-second sample from these jars, and we were getting chemical information in less than 300 seconds after that collection time. So you can see here where, you know, I don't think potentially in terms of a sorting facility, you know, I've seen some of the imaging where, you know, in a matter of nanoseconds, they're able to get a reading and sort based on that. But certainly here in less than a minute, or certainly less than 10 to 15 minutes, you can get some answers and get an answer to the question of interest here. So this is where we see it as especially maybe useful in some sorting or packaging facilities, again, where you're maybe wanting to assess the quality of certain containers, for instance, where you have time to go through.
Banana example, how would you view it actually being used commercially? Is this used commercially?
This idea is not being used commercially at the moment. But what we foresee it to be used as is a VOC sensor that gets built into the shipping container, and that it's constantly measuring samples. So whether that's at a frequency, you know, I don't think at that point you need minute by minute, you know, resolution, but certainly every 10 minutes, for instance, you're taking a measurement, and that provides information to the control system about whether to engage refrigeration or not.
And would those containers of banana be dedicated just to bananas, or would they be entering the pool of containers around the world? That would be the other thing, right, in terms of, you know, retaining the technology for that, specifically for that industry.
Yeah, exactly. So it would of course, depending on the applications. So you know, this particular sensor would be trained on banana ripeness. I would hope that it could also be used for some other things as well. But to your point, yeah, actually we have Erwin and our group have a pending proposal together to look at some detecting pests inside shipping containers as well. So.
It's really interesting to see that the dog can, like, you know, sense the HLB. I was wondering if it's possible to, like, develop a sensory panel of dogs who can do the wine testing or other things. Like a dog tested certified thing.
Yeah. I mean, there's so much potential for dogs. We see them, you know, we see them being able to alert people who you know, are managing blood sugar levels, who might need to be alerted if they have an upcoming seizure. You know, they've been trained to be able to identify certain lung cancers. So dogs are really showing the ability of using volatile compounds there as a VOC detector. Again, they're just expensive to train. You know, there's a lot of handling needs for them as well. So, you know, in terms of high throughput, it may not necessarily be an effective way for the industry for them to be used there, but I think they could certainly accomplish some of those, sure.
I'm going to add to him. I think if you look at the price of equipment, and the equipment does age faster than dogs. And the dog can actually detect can sample faster than the equipment that is being developed.
Absolutely, yeah.
I think that if, for example, the state of Florida had maybe 10 dogs, they put money to training 10 dogs, they can finish the whole state. Maybe on a monthly basis, because for 7 months you start detecting that. So those dogs can do the job faster than the equipment. So I really agree with the idea of developing for the dogs, and then we'll waste less money in that area.
Of course, but I think, you know, some ideas with the VOC sensor is that, you know, you can't put a dog on a drone and fly it around. So, you know, in terms of screening for pathogens, you might be able to do some high-level work where you take a VOC sensor and sweep it through fields and start to see if you see any evidence of a pathogen or a pest or whatever it is you're measuring. Maybe at that point, you go back through with a dog or a VOC sensor and start to fine-tune exactly, well, which tree is it that's infected? In the case of Florida, unfortunately, all the trees have HLB now. No one is actually worried about diagnostics because they're all either infected or will be infected. So there's not exactly, unfortunately, a need for that right now in that state.
My other question was regarding the walnuts. That you sample right they want to work from lab.
Yes, thank you.
How did the scale from 1 to 7 actually what you found?
Yeah, so that's the ongoing work we have now here, so this was using some chemometric techniques. We knew the grade of the walnuts that were provided to us. So we trained the walnuts on individual grades, and then we fed in samples where we knew the right answer, but we wanted to see what the model predicted. And that's what we're comparing here, was we had examples where these models were able to, for grades 1, had 100% accuracy to predict what samples were grade 1. Same for grade 2, 100% accuracy. And right now, again, this work is ongoing, but as you move to more rancid walnuts, this particular model had a harder time to correctly identify which grade.
Therefore, it means that those grades were given by eating the walnuts. Wasn't actually the nose it was eating, and it's sensing, when you are eating them, then you know which one were rancid compared to the ones that were not rancid. When you get to the level of 3, 4, and up, that's when it gets confusing because your body can't actually tell the difference.
Right, which is where, it may not matter that the model has a harder time to distinguish more rancid grades of walnuts because they're already at such a deteriorated grade. Who cares, right? Yeah, exactly. But certainly, for produce quality, here you have a premium product, right? And again, I'll be here for the panel discussion in case we don't get to anything.
All right, I can read some of the online questions. So Medina is asking if the GCDMS is commercially available, or if it is, then any recommendations for suppliers? Yeah,
it's a good question. The original manufacturer of the GCMS we were using was a company called Cyanex. They actually had a plan for closure. They didn't survive the recession in 2008. But I think it shows the quality of the product because we're still using these devices in our laboratory. So they potentially are living at least as long as a dog, if potentially not longer. So our group is actually now licensing technologies through our affiliated startup company, and with funding from the NIH, actually developing some new commercial products. So there are a few other vendors that exist that use ion mobility spectrometers, but again, they're not tailored for any specific VOC sensing application. They're more just general VOC detectors.
So Anna is asking, how close does the GCDMS suitcase need to be to the infected or healthy cultivars to detect VOCs?
You said how close to the trees?
The cultivars, like if you have
the cultivars
or something, how close does the GCDMS need to be for you to be able to detect the VOCs? Yeah, and that's again really depending on the exact plant species. Some produce a lot more odor compounds than others. In the case of citrus, they smell great, and they're pretty large, so it's actually pretty easy to get a sample of them this way. But this application wouldn't work for every single commodity crop. You can imagine some smaller crops. There's just not enough plant material. Melons growing in a vine, for instance, they're not kind of all concentrated, and all the plant tissue isn't concentrated in one area. It's a little bit more harder to measure a volatile sample of that. So it really can depend. I guess one last question from our online viewers. Can you share more details on what has been done for cacao?
For cows,
for cacao.
Oh, cacao. Yeah, I did have a photo of that. I actually, a friend of mine with Professor Sue Ebler She did her master's thesis looking at some VOCs related to cacao quality. That was actually looking at a study about how cacao was being grown, since cacao typically actually prefers to be grown under shaded conditions. In a lot of industrial cacao production, they're out just kind of exposed direct to the sun. And using VOC sensors, we showed that direct sunlight actually generated acetic acids and other compounds that really kind of gave an off flavor to the cacao. Well, later in the production of chocolate, I should say. So here you can also be using a VOC sensor up front in production, pre-harvest, to be able to kind of predict the quality of the product post-harvest.